quiero recordarles unas instrucciones de seguridad en caso de que algo ocurra. Vamos a evacuar el auditorio, la mitad para allá y la mitad para acá. Las puertas ya están libres. Los hallways deben quedar libres también, sin ninguna obstrucción. Por favor, apaguen sus celulares o pónganlos en modo de silencio. Bueno, es un placer para mí presentarles al Dr. Nicholas Priest. Él lidera un laboratorio que estudia el género de moscas drosófila en el Centro Miller de Evolución en la Universidad de Bath. El Dr. Priest obtuvo su doctorado en la Universidad de Virginia y continuó su entrenamiento con un postdoc en la Universidad de Indiana. Él ha hecho contribuciones importantes al entendimiento de las compensaciones o trade-off sobre la longevidad, la dieta y las infecciones en insectos. El Dr. Priest también es un comunicador muy entusiasta y ofrece charlas a varias escuelas, en ferias de ciencias para chicos y en eventos para adultos también, como el famoso Pint of Science. Y bueno, sin más preámbulo, el Dr. Nicholas Priest. Bueno.
First, I'd like to say thank you very much for having me here. It might seem awkward to carry the microphone, but if it's being recorded, then I need to use it. So the people at home, hi! If anyone ever watches this, can listen in. So let's see here. There's Lisa, and let's see. Oh, there. Yes. So I should point out that we have several goals for this visit. It turns out this is my probably fourth time visiting here, and in fact my second time visiting this year. And it's been a big effort has been made by my dear colleague Araxia Lucia, who has been encouraging all of her colleagues at Bath to consider research collaborations here at UNA. And what I've seen in the many visits I've had now to Ecologia and now getting to know Biologia better is that the potential for synergy across the pond is tremendous. And in particular, there are a new set of grant funds that are meant to accelerate international research, particularly on problems that matter, that have big impacts in society. So, and it's been great meeting academics here who are like-minded and wanting to solve big problems. Well, in addition to basic research and finding out mysteries that may not solve problems but make important contributions to science. So, in terms of the reasons why we're here, I'm interested in research collaborations relating to behavior and genomics and Chagas disease. Alex Cordova from Ecologia has been instrumental in helping us develop plans here. But I should point out that we're developing collaborations across Latin America, particularly related to Chagas disease. And for those of you that are interested in these kinds of international collaborations, I should point out that it's not just all meetings. There is actually a lot of fun involved as well. So this is cycling in Bath, in the UK, through the countryside, very nice, and also events. This is the famous Roman Bath in the city of Bath, which if you ever visited Bath is probably the major tourist attraction. And I should point out that we're very interested in having PhD students and postdocs, and even technicians for short visits. These are relatively easy to arrange. And in particular, Araxia has been instrumental in trying to encourage more collaboration with PhDs. In fact, I think there is currently a proposal being considered for a joint PhD program from University of Bath and UNA. And I should point out something about Bath. we're not that big of a university. In fact, we don't even show up on the international league tables because we're very small. But the group in evolutionary biology, in particular, is outstanding. And the students that have visited us, I think we've had now probably 25 visits, and it turns out even Alex has mentioned that his students, when they visit, they come back charged, ready to go on their PhD. So they can be good experiences for research. And mainly, we really want to solve big problems. That's our goal, through collaboration, solving problems. And I should give a special note here to, this is a visit, I think 2 years ago in Cuernavaca. But I do want to, in particular, mention my close colleague, Araxi Rucha, who many of you are probably aware has accepted a position here. She's leaving Bath, although we're very hopeful that she will be willing to keep a small appointment so that we can take some credit for her work. But Araxi is really at a crucial moment because she's developed methodologies relating to transcriptomics across time that are really cutting edge in the field. And so she's coming here at basically a sort of the peak in terms of her abilities, and there are so many colleagues at Bath and also people that have come to Bath that have been super excited about collaborating with Araxi on various projects. Yeah, so this is just to point out, we've had lots of visitors, particularly in recent years. So this is Martin, who it turned out, Martin was a, did he do his PhD?
He was a PhD in Bath.
Yeah, he did his PhD in Bath and is now at Puebla. And I should point out, this is a walk in the countryside, not too far from other. This is a burial site that's older than Stonehenge, and it's sitting out in a field in the middle of nowhere. And this is part of the fun of living in the UK is the history, just like the amazing history here in Mexico. So much to discover. And again, it's also fun at times. This is a colleague, Sam Shepard, who's been very excited about developing more collaborations. He's an expert in the genomics of pathogenic microbes, hospital-associated, and has been very interested in developing links to access collections here to ask basic questions about how virulence evolves. And I should point out we have developed now strong collaborations with academics from throughout Latin America, but especially in Asunción. And I should point out, yeah, so this is Antonieta de Arias, who is an expert in Chagas disease, and in fact, she and I have just won a grant to run a conference next July with all of we're hoping to attract many of the experts in Chagas disease. And her friends are amazing. Most of them have 200 papers on relating to Chagas disease. And so the hope is with this grant, we can help raise the appreciation for the excellent research that's happening and develop a strategy for attacking Chagas disease. And so we've now done, or particularly over the last, it's only really been a year and a half, 2 years, we've had many visits with Paraguay and Mexico. We've developed a network of collaboration throughout all of the countries affected by kissing bugs. We've received substantial pilot grant funding from the UK government, and we are. It's my sort of consideration of the field is that actually what we need for Chagas disease is more people to know about it, and we need the UK in particular has expertise in public health and climate research, basic entomology, disease ecology. That I think if we can make those links, then we can develop a more effective strategy. There are 10 million people at a minimum affected by Chagas disease. And it's one of those diseases we just don't know enough about to be able to make an informed strategy. Okay, I should also point out that part of the reason to make these efforts now is that at the University of Bach, there's been a big interest in our research group. And in fact, the Milner Center for Evolution is a brand new research facility just for our research group. And this is now, this is part of Martine's, this is a different visit from Martine, actually. There he is inside our space. And again, I should point this out that on my last visit here, we examined the insect collection for Triatoma. And we've been hoping to establish discussions so that we can study this collection in further detail, because I think it has impacts on understanding the spread of Chagas disease throughout Latin America. So these are the Triatoma that cause disease, and actually some Triatoma that don't. Okay, so now my core research relates to how animals fight infection. And I think that some of the problems with why Chagas disease is so complicated is that when the kissing bugs carry Trypanosoma cruzi, they are actually fighting the infection. They're changing their internal physiology. They're not just inert, they're acting in a different way, in an unusual way for them. And so it makes sense then to think about, okay, so what do we know about immunity, and how does this relate to how insects behave? Now it'd be nice if we could do this in a very detailed way for kissing bugs, but sometimes it's also useful to use a model system to study it. And in fact, much of what we know about immunity has come from research from the fruit fly, Drosophila melanogaster. And so what we've discovered is that there are immunological pathways involved. So things like encapsulation of microbes, melanization of tissue, and a whole set of signaling pathways that are initiated whenever animals get a big infection. Now this is sort of the state that's considered to be kind of the state of the art of the field, but we need to study these mechanisms in more detail to really know how they fight infection. But I've also been looking at kind of the dark side of immunology. And in my lab, we've discovered that animals show responses that are not considered to be part of the canonical immune system. They act in ways to fight infection that at first sight seem bizarre. So they exhibit anorexia. They stop eating, or they eat much less. Fruit flies seek out cold climates. They also medicate with protein, so you can show with behavioral tests that they'll seek out a higher protein diet. And then they act in ways that you would think for immunity don't make any sense. If immunity is about surviving infection longer, then why would they boost up the production of eggs? Now, in an evolutionary viewpoint, of course, it makes sense. Reproduction is the key here, right? That's how evolution works. But the immunity field doesn't consider reproduction as being an appropriate response. It should only be about survival. And when I talk to the top guys in the immunology field, in fact, Jules Hoffman's lab, they've all said, oh yeah, it was a political agenda, so that when you think of immunity, you think of Jules Hoffman. And that hasn't been a consideration. Okay, so how do animals actually fight infection? So what I want to argue here is that actually what's critical is that it's actually all about nutrition. And what I want to propose is that, contrary to think about immunity being how animals fight infection, really it's about the gut. And in particular, when you work this out in further detail, what the implication is that evolution is constrained by protein and protein use. And so the molecules animals use to fight infection are limiting to some extent. And we have to think about how the microbes within the gut are using nutrients in addition to how the hosts are using them. So, now, so that's the big question, is about, does gut trump immunity? Is gut more important than the immune system for fighting infection? Now, tengo una palabra. So this is so I want to start with a little diversion here now. Just a kind of general question for the audience here. Evolutionary research has produced lines of fruit flies that live extraordinarily long through artificial selection. So the classic aging studies done by Michael Rose and Brian Charlesworth in particular, but lots of other people. Arkin and Luckinbill, and there's a whole series of famous fruit fly lines that live a long time. So the question I want to ask you is, how do they get this long life? How do fruit flies get this long life? Or do they get long life by eating less? Do they get long life by changing their food preference? Maybe they're just better at finding diets that give them long life. Do they lengthen their telomeres? Do they make the gut less permeable? So this is a little bit of a, for those that aren't in the Drosophila community, this probably won't make as much sense, but there's been a lot of assays on gut permeability, like how the gut degrades when challenged. And then the other one is, how does it change through how they process nutrients? Okay, so what I want to do is to take a survey of the audience here. And you can choose multiple choices if you want. Do you think fruit flies eat less? I mean, it's the major way that there have been interventions in longevity and across multiple species. So it's a good answer. Okay, so we got a couple there. Do they change what they eat? Okay. Do they lengthen their telomeres? Okay. Yeah. Yeah, so particularly in humans and yeast, and in many bird species, this is a well-documented phenomenon that the telomeres shorten with age. So that's a good possibility. Gut permeability, okay, so that's a classic one. And then nutrient processing, okay. So basically we did some investigations to work out for these long-lived flags. Let's run assays, let's see which ones work. But I want to start with this, that in some ways, even asking this question is unsatisfactory. Because what we need to know is not just the mechanism, we really need a biophysical explanation for why those things change. So if telomeres decline if they've grown longer telomeres, how exactly are they doing that at a molecular? What molecules are they using? And what does that cause a life history constraint somehow on a molecular level, at a biochemical level? So that's what I mean about a biophysical explanation. We should take it down to first principles if we're really going to have a full understanding. Okay, so we've done a whole bunch of tests to work this out. So when we have conducted nutritional geometry, so this is a process by which we put flies in tiny tubes that have even tinier capillary tubes inside them, and then we alter how much protein and carbohydrate we put in each tube. And then we, just using calipers, measure how much liquid is missing. We also do things like lots and lots of controls to make sure, well, it's not evaporation involved. Actually, turns out there's always evaporation you have to account for, but then by very carefully altering the level of protein in each tube and the level of carbohydrate, and then using dilutions to make it so that they actually have to eat a lot more to get the same calorie intake. We can then repeating this lots and lots and lots of times for different nutrient ratios, and then mapping on top of that for thousands of fruit flies, how long they live, or how many babies they have, how many eggs they lay. We can then map on a three-dimensional surface the relationship between a nutrient intake and, for example, the trait lifespan. And sure enough, if we do this, it turns out that effectively a high sugar diet with probably pretty low protein, but mostly just a very sugary diet, gives fruit flies the longest life. So this is where the reddish tone here, these are the longest lived flies. And I should point out the model doesn't say that it goes up and down. Actually, basically the more sugar they eat, the longer they live. Now, wouldn't it be great if that were true? And I should point out here that when we then test, okay, what do flies actually choose, using a whole set of different ratios in a fairly rigorous methodology, it works out that they don't actually choose diets that give them the longest life. But the take-home message here is that when flies are healthy, sugar is good and protein is bad. And this sort of leads to the question, okay, but what about when they're infected? Do they actually need, does protein need then shift? And then if we're thinking about biophysics here, maybe protein is the molecule that's really limiting in the sense of it's bad for them when they're healthy, but they really need it when they're sick. And so, and to do this, we've done these experiments in standard lines, and we've also done them in these long-lived lines. So this is the famous Bob Arpin line. And essentially what we did was we took flies, infected them with Metarhizium revertii, which is not a pathogen of Drosophila, but is a common entomopathogenic fungus. It's kind of a model entomopathogenic fungus, often used in ant research. And essentially what we do is we just drop fruit flies in a flask that have the spores in them. And we had healthy controls. And we had lots of cages, and then measured their survival over time, and also took collections of how they ate foods, and also we collected poo from these fruit flies, and then did biochemical analysis on the poo to work out of what they ate, how much did they actually process. And then we also did assays on their diet preference. We looked at telomere length, and we looked at questions relating to gut permeability and other things. So if you just look at how the flies live, when they're healthy, this is on a standard fly diet, it turned out the longevity selection still works. Even though Bob Arkin did the selection in the 1950s, they're still living longer across time, the longevity selected lines. But when they're healthy. But when infected, the longevity selected line crashes. Mortality goes way up, and much earlier than in the control line. So they're much more susceptible to infection at an early age, these long-lived lines. And the long-lived lines, but however, if you were to then collect the flies, they're healthy until day 40, and then you apply the infection, it turns out that they're better at fighting an infection when they're older than the control line is. And again, you see they're still, in terms of the longevity selected lines, yes, they're still living longer at day 40. So longevity selection, yes, it improves survival, but it decreases susceptibility to the infection at an early age. So when we then did the rest of it, the nutritional geometry, we didn't find that the longevity selected food preferences. There were no changes in telomere length, and no changes in gut permeability either. So that one of the last questions was, well, what about protein processing? Now, if we were to look how much they actually ate, what we find is that the longevity selected lines eat the same as the control line at that early age, and they actually eat a lot more, just total food, what do they eat, than they eat a lot more than the control line at a late age. And then we work out, okay, well, how much of what they ate did they actually use? And at that early age, the longevity selected lines are pooing out in their frass. There's 150% more protein than they ate. So they're shedding protein. Whereas it's still a lot of protein being lost by the control line, but much less of what they still absorb much of the protein they're eating. So in general, flies don't have perfect absorption of the protein they eat. Typically, for most lines, it's around 70% or so of the protein they consume will actually be broken down into uric acid. And then later in life, the flies are better at absorbing protein. So basically, these longevity selected flies are altering their protein processing. They excrete more unprocessed protein at an early age. And then it's better later. So how do long-lived flies live long? It turns out all of these questions, they don't eat less, they don't change their food preferences, all of these, we find no effects, and any process we find is about nutrient processing. So there is, long-lived flies are actually excreting much more protein than the control lines are. Okay, so that's sort of an interesting, this gave us some insights into how animals use protein. This is sort of then, are they somehow altering their guts in ways to fight infection? Because if they don't process protein correctly, then they're more susceptible to infection. So this could be a mechanism by which animals fight infection. Just alter how much of what you eat do you actually use. And I should point out that if you go back to the history of immunology research, most people use high-dose infections in their studies. There are very few labs that give a very light touch infection or low-dose infection. And most of the immunity research that's done in flies is about how many hours do fruit flies live after an infection. And if they have or don't have an immune gene, then they might live a few hours longer. Whereas where low-dose infections are much harder to do because you're looking at very subtle changes in their survival. So maybe that's the reason why they never focused on this, or maybe actually the thought was a large dose infection will give you inferences for how they fight the typical infections they face every day. And again, Jules Hoffman has been a big part of the problem, is that he said, no, the assays have to be done on high-dose infections, that's the standardized methodology. And in the field, actually these low-dose infections have a hard time even getting through reviewers' studies that include those. Okay, so one way we can do this is study both large-dose infections and a low-dose sexually transmitted infection. Or other kinds of low-dose infections. And then work out, okay, so for these different types of infections, how are they altering protein processing in the gut? And what we did is we worked with the biochemistry lab to develop a fluorescence assay that then allows us to compare how much healthy protein processing is there compared to the unhealthy, unprocessed protein. And with a single fluorophore, we can get two peaks, one that fits protein and another that fits uric acid. And so then by taking a mixed sample, we can then look at the ratios of the peaks to deduce the relative amounts of one or the other. And when we look at naive animals, so untreated animals and collected poo and ran the assay, or STI animals with sexually transmitted infections where there is a kind of bit of a double bump there, and DTI flies, and then calculated the relative amounts of protein and uric acid. What we see is that this is the average ratio of the two, I guess. What we see is that for the naive flies and the DTI flies, you actually see pretty similar ratios. But for the STI flies, these are the low-dose infections that don't really affect survival very much. What we see is they produce a lot more uric acid and a lot less unprocessed protein. So this shows that when animals contract a sexually transmitted infection, they alter their digestion. And this led to the question of, okay, so since STIs is the kind of low-dose, subtle infection that alters, and we could show that it alters gut processing, then that suggested, well, since we have this interest in non-immunological mechanism, could this be the type of infection that where flies could seek out medicines in nature to try and adjust their digestion? So maybe with a high-dose infection, they only live a couple hours, so they don't even bother seeking out nutrition. Maybe with these low-dose infections, they do. So that led to the question of what kinds of natural medicines should we be looking for fruit flies to go to? And how does this work? Is there a different kind of mechanism than the immune system? Is there a non-immunological genetic mechanism? And then how does this kind of behavior evolve? So an obvious one is to think that this is Drosophila, right? The fruit-loving, is to consider the natural yeasts that inhabit rotting fruit. And these are different, so we typically, I don't know, many of you that work with insects might use brewer's yeast as your typical yeast source, pretty high in protein. These natural yeasts produce a whole range of compounds that are attractive to insects, including 2-phenylethanol, so an alcohol that has a benzene ring. So this seemed like, well, if we're thinking about molecules that are produced in high abundance by these natural yeasts, that could be a molecule that maybe the yeasts are producing this to get the flies to eat it, sort of in a multi-trophic kind of mutualism. And so one thing I should point out about flies is that before they have sex, there is a fairly protracted courtship period. And males in particular use song. So males play songs, and this has been shown to induce a whole series of physiological mechanisms that are basically do result in the remarkable transformation of a virgin female into a female that's a fully inseminated female that's cranking out eggs at a high level, eating a lot more, and basically internally having a major shift. But even song can do that. So that got us thinking, could we play song to females before sex and then test whether they drink this alcohol? Could we use that as an assay? So this is pre-drinking, before sexual intercourse. And maybe they're doing this to protect themselves from the infection they're about to get from the male. And so we did these experiments, and sure enough, that's what happens. If you play the mating call of Drosophila melanogaster for females that have 2 tubes in their cage, one that has 2-phenylethanol at a low concentration and one that's with alcohol, what we see is they all drink the same amount of alcohol, but when they get the call from Drosophila melanogaster, they drink more. If you give them the wrong call, they drink less. And if you give them no call at all, they will still drink the 2-phenylethanol, but not as much. And this then said, well, maybe that's a good molecule then to test for whether it actually helps them. And so what we did is we did experiments where we took females and gave them sexually transmitted infections or large dose infections or no infection, and then measured, okay, how does that affect their longevity? And when we, on diets with 2-phenylethanol spiked in it, the females live longer. So they get a longevity benefit if they have an STI and they're drinking this alcohol. Now crucially, for it to be a medicine, by definition, medicines are bad for you when you're healthy, at least in the sort of classic definition. And what we find is that 2-phenylethanol is costly to females when they're healthy. And what we find is actually this drinking alcohol does not help the flies when they're given large dose infections. And I should point this out that it's the same microbe, it's just delivered in two ways. We coat fly females in the spores, or we coat their sexual partner in the spores and allow them to mate. So it's specific to their infection mode. And one of the craziest results we found was that 2-phenylethanol, when they did the same test for what is the ratio of uric acid to total protein, what we find is that 2-phenylethanol improves the digestion of protein. So now it raises from 70% to 90 to 95% when they have an STI and they have the alcohol. Okay, now in terms of mechanism, we did the same trick. We played mating calls to females, chopped off their heads, and then used transcriptomics to work out what genes are expressed in the head of the female. And we found that that the most upregulated gene in the head of a female when she hears the mating call of a male is this gene torrendose C. And then we then used immune gene knockdown lines of fruit flies to work out, okay, what does this gene actually help them drink alcohol when they have a mating with a dirty male, when they get an STI. And in the naive case, no difference. Large dose infection, no difference. But in the STI condition, we see a shift in their feeding. And when we work it out, when top C is knocked down with the UAS Galfor system, they drink substantially more alcohol. This is on the first day post-mating with a dirty male. A female will drink substantially more alcohol than either of the control lines, so where they have the antisense vector but not the Gal4 to stimulate the expression of that, or when they have the Gal4 but don't have the antisense vector. So this says then that TOTC regulates or influences the consumption of alcohol after having a sexually transmitted infection. And in terms of thinking about the evolution of non-immunity, what we've discovered is this TOTC gene results in reproductive trade-offs. Now, and I just wanted to add a kind of question about, okay, so when in general does immunity help? and is, just when we're healthy, is the purpose of our immune system to help us fight infection when we're young at a cost to our late life, to our health in late life? Or is it actually, is what we call the immune system not about fighting infection per se, but actually it's about helping us live longer, providing protection in late life? Now, as you say, evolutionary theory would suggest really it's all about early factors that matter. If you can start producing your babies early, then that is more likely to give you a competitive advantage. And late life, gene expression that helps you in late life shouldn't work, particularly in an increasing population. It doesn't make sense for immunity to help in late life. Okay, but this is the kind of thing we can test with fruit flies. And so what we did is we took immune genes and then measured how they aged using large-scale demography in combination with demographic modeling. And so what I'm showing you here is the log of the mortality rate across time for a set of control lines and for the immune gene knockdown line. And so this straight line here, this is referred to as the Gompertz curve. And it turns out this is the same curve insurance companies use to work out what your premiums should be. You go over a prediction of your own death. That's what they use, it's still the Gompertz curve. But in severely immune deficient animals, animals where they've had the diff, the central diff gene turned down, this is the mortality pattern we see. That mortality rate is low in early life and then increases late life. Now the significance of this is that just focus on what's happening at this end. You find significantly different shift in the rate of aging, the term here that expresses increase in mortality rate over time. The significance of this term being different is that this is saying that immune genes tend to reduce when they're functionally active. They tend to reduce aging that happens, the increase in death that happens. So basically, you get rid of the immune system and you age much faster. So basically, you get late help from an immune gene. And then we looked across a range of immune genes, and we see the same thing in relish, and we see the same thing in another stressed immune gene. We don't really see shifts in the rate of aging in late life. This is these with stress-related proteins. So in this case, actually, although there's a B term there, it's actually the slope declines. And in another stress-related type C, we don't see that. So because it's not significant, but the point is, in general, with these immune genes, ones you'd say are canonical immune genes, particularly diff and relish, you see very much late help involved. And typically when people have looked at these immune genes and seen, well, you get rid of the immune gene, like diff and relish, and you live longer, then that's an indication of immunosenescence, or they're living longer, because the immune gene is helping them in early life. But there's another argument that it's about late age protection. So when we look at infections across time, we don't see evidence that these immune genes are hurting in late life. And that's, when you do lots of assays with fungal virulence, what we see is that actually as animals get older, I used to read this, their hazard ratios normalize to early life. Animals get better and better and better at fighting infection as these curves are going down. And when we look at immune genes, and we just plot this according to changes in mortality using our demographic modeling. In general, we see late health, that the difference in mortality rate in late life is when you get rid of these genes, mortality rate goes up in late life. And we find similar things when we challenge animals with fake fungal infections, late health. So when we think about how animals fight infection, we now have established evidence that it turns out that animals use natural medicines. They employ very different mechanisms than immune mechanisms. And they exhibit the kinds of trade-offs we would expect for evolutionary phenomena, ecological immunity phenomena. And then when we do tests of when immunity helps, it turns out it's in late life. So I started with the claim that actually we want to consider how animals fight infection. It really there is a question of whether it's through the immune system or actually through shifts in the gut. And effectively what this means is alternative medicine works. The fruit flies use alternative medicine to fight infection. They don't use their immune system And in fact, they use their non-immunological system. But when we do closer dissection of what the molecules involved, it appears that protein really is this constraint. It's good for you when you're sick, but bad when you're healthy. And that use of that molecule then constrains life history. And I think we're doing studies to work out whether this is just a kind of fundamental biological explanation for evolution. And it turns out we have evidence that protein absorption affects basic evolutionary phenomena like meiotic recombination. And we suspect that protein use also influences things like mutation rate. So through this work, we're hoping to make new basic inferences about how evolution works, and also ones that relate directly to solving the big challenges, health challenges facing the world. So all the work we're doing right now is on looking more detail at temperature seeding, and so we can say very clearly that when flies move to the cold, they are shifting their intake of cold nutrients. So it is gut related, that cold seeding. And similarly with anorexia, you interpret anorexia as, oh, they're eating less, but it turns out when you give flies a choice of foods, they seek out high protein diets when they're eating less. So it actually is related to protein use. And we're doing assays working out how amino acids can mitigate against infection, so particular nutrients in the environment and how they can help animals fight infection. And of course, I'm sure this is on all of your minds. The big question is, can we fight infection and stave off old age by drinking red wine? And I certainly hope the answer is yes. Thank you.
Okay, tenemos 15 minutos para preguntas. No, no hay tiempo. No hay tiempo para preguntas. Bueno, yo tengo una. Yeah, regarding the STD infection that you gave, well, so by definition, that's a sexually transmitted disease, infection, right? So they have to mate.
Yes.
My question is, besides music, flies also use olfactory cues,
Yes.
right? So the question is, do healthy flies somehow, through olfactory cues, can mate with a sick one, or do they prefer to mate with healthy ones more often? They are definitely mating, because there is transmissions.
Sure. Yeah. So it turns out that they prefer dirty males. Go figure. And it turns out females that have mated with a dirty male also mate more rapidly with the next male. And it turns out males, on the other hand, are more likely to spread infection. We have not looked at olfactory cues and these anticipatory cues. I think actually could be a really exciting way for further advancement of genomics, so working out how animals are perceiving these cues, and particularly ones that predict infection. I think could be very fruitful.
Very nice, Dr.
Well, thanks.
In the general diagram where you have the female and male describing about the courtship, you indicated that it also affects fecundity.
Mhm.
I would wonder if you could go a little bit further into
Yeah.
that.
Yeah, so the typical pattern we find is that when females mate with a dirty male, they produce more eggs immediately. Sometimes you only see it in the first 24 hours post-mating. And it turns out if you mate with drinking, then there's even more egg production. And in general, having alcohol around creates more babies. I mean, this is preferred flies too we're talking about here. You know? But then when we did a dissection of the TOC gene in this, it turns out that TOC regulates egg production directly in response to infection. So we think we have evidence that TOC is the gene that controls fecundity compensation, or one of the genes that is at least involved in that. So yes, we're finding high reproduction immediately post-mating with a dirty male, much higher than the control mating with control male, and it's influenced by the expression of the TOTC gene. And in general, TOTC appears to have a cost to the longevity of the fly. But again, it gives a help to the reproduction of a fly when she mates with a dirty male. So it kind of makes sense in an evolutionary framework. It does not make sense in an immunity context. This is certainly a non-immunity problem or an eco-immunity problem.
Well, I have a question too. I mean, it has been shown that a lot of parasites, they actually can change the behavior of the host, and this is in fact for their advantage. So, I mean, when you observe all these things, how much do you think is actually good to fight the parasites, or do you think it could be some kind of evolutionary process which is actually to enhance the disease?
So in our cold-seeking behavior paper in Journal of Animal Ecology, we did a lot of work to work out, okay, how is this helping the parasite? I should say a lot of studies that look at host manipulation often don't include extensive sets of controls to know, well, what is the preference of animals normally? So for example, when flies, it's always been said flies move to the cold or move to the hotter temperatures when they're sick to improve their survival. And sure enough, you can show that. But actually, usually you can show that's true for control animals as well. And when you work out the fitness costs, the fitness benefits to the host, if it's infected, that was sort of we really wanted to work out. Is it costly for the fly to move to the cold temperature? Is it costly for the so is that a manipulation by the parasite? And it works out in the tests we've done. There are major fitness advantages for the host, but it's sometimes a bit bizarre. So by moving to the cold, the fly reduces its intrinsic rate of increase, the instantaneous growth, but it gets higher lifetime reproductive success. So we interpreted that as actually they're doing this to get more babies, they're just doing it late in life. So I think it's actually very tricky to work out who's being hurt here and who's benefiting, but certainly the host is responding, and certainly in ant literature, there's much better evidence of manipulation of the parasite to the detriment of the host, but often it's neutral, whether it hurts them or hurts the host or helps the host.
Bueno, si alguien más tiene preguntas, pueden hacerlas durante el café.
Thank you very much.